Hello students. We are discussing on the primary and secondary structure of protein. And now proteins are divided into the four classes: primary, secondary, tertiary, and quaternary, on the basis of their configuration and orientation in the space. Now, first of all, we are concentrating on primary structure and secondary structure of protein. Now, the primary structure of the protein, what is it indicate? Now, primary structure of the protein is a linear structure. It is a linear structure. It is a linear structure, and it is a sequence of amino acid. Now, one amino acid is attached with another amino acid and it form a long chain in the space. This structure does not have any bonds like hydrogen bond, covalent bonds. Covalent bonds are there in between, but there is not a interchain covalent bond. Now there is a van der Waals forces, and there's an ionic interaction. Now this is the primary structure. The example include insulin. Now, in a secondary structure of the protein, it is a folding of a chain in a space with help of hydrogen bonds, ionic bonds, van der Waals forces, these all bonds act in a protein structure and form different shapes. Now, secondary structure it include two form. It is one is a alpha helix and one is a zigzag pattern like structure, which is known as beta pleated sheet like structure. Why it is known as beta pleated like structure? Because in the space there is an orientation like this. There is a plate like this which are oriented in a space, and this one will form a one plate, and this down linear chain will also form one like this plate like structure in 3 dimensions. So it is a beta pleated sheet like structure, and it is alpha helix. Now primarily we are discussing on this in a secondary structure. When we discuss about a secondary structure, we are primarily discuss on alpha helix and beta pleated sheet. Now in alpha helix, it is the alpha helix. It is the most common available structure, most common secondary structure, and it is the most stable. Now it is a right-handed structure always, and And each turn, this one will make one turn. Each turn is made up of 3.6 amino acids. 3.6 amino acid, and now 3.6 amino acid is present in one turn. And now there will be for the stability of this structure, there is a responsible there is an intra-chain hydrogen bond like this. Here intra-chain H bonds are responsible for their stability. Here in this turn, first amino acid and fourth amino acid will form the hydrogen bond. This is an intra-chain hydrogen bond. But now there are the various type of amino acids. They are present in this alpha helical structure. We are discussing the amino acids. Amino acid which are present in alpha helix. It include methionine, glutamate, alanine, and leucine. These are the amino acids which are present in alpha helix. Now, there are certain amino acids which do not favor alpha helical structure. These include beta amino acids. These include amino acids having branched at a beta carbon. For example, this is CaOH, C or C. The carbon adjacent to this group is alpha carbon, and this is the beta carbon. If here there is any branch or methyl group or ethyl group, then it is a branch amino acid. So some amino acid having a branch with beta carbon, they will destabilize this alpha helical structure. This amino acid will include isoleucine and alanine. Valine and threonine. Isoleucine, valine, and threonine, when they are present in this alpha helix, they are destabilize this structure, and they do not favor this configuration due to this branching present on beta carbon. They do not favor this type of helical structure. They were always absent in this alpha helical structure. Now there are certain amino acids which are disrupt the stability. Now this amino acid like isoleucine, valine, and threonine, these amino acids does not favor this type of configuration. They are always absent, and we can say these are always absent in this alpha helical structure. And now there are the certain amino acids which are destabilize destabilize alpha helix. This will include serine. When serine is present in this alpha helix, it will always destabilize this alpha helical structure. Serine, aspartate, and asparagine. When these are present, they will always disrupt the stability of alpha helix. Now, proline is present in alpha helix. Proline is amino acid which is present in alpha helix, but in a small amount. This proline, it is an amino acid. It is present in alpha helix in small amount. It is also disfavor the stability, but not like that. This type of amino acid, they are always destabilize the structure, but it is proline. It is in a small amount. Proline is present in small amount in alpha helical structure, and this will also disfavor this alpha helical structure, but it is very little. There is little disorientation in the structure. While this amino acid, when they are present, they always destabilize the structure. Serine, aspartate, and asparagine. Now, glycine is amino acid, which is not glycine is amino acid, which is anyway they do not favor अल्फा हेलिकल स्ट्रक्चर ऑलवेज. आप इसको कुछ भी करो लेकिन ये अल्फा हेलिक्स नहीं बनाएगा. Now in beta seated structure is it is the orientation in the space. They may run parallel or may run anti-parallel with each other. Two configurations are there. Parallel and anti-parallel. In parallel, the two chains are run in the same directions, while in the other form, it is run in anti-parallel direction. Example of parallel configuration include flavoprotein, flavodoxine, and in this example include silk fibrin. In flavodoxine, the beta-pleated sheet-like structure is in a parallel configuration, while in a silk fibrin it will be in anti-parallel arrangement. Now, this beta-seated structure they are stabilized by interchain H bonds. Means the hydrogen bonds are formed in between the chains. These are the interchain hydrogen bonds. While in the alpha helix, hydrogens are bound in between two amino acids which are present in single chain. So these are the intra-chain hydrogen bond. Word intra is used. While here there is word inter is used. Now amino acid like valine and isoleucine are most common in this beta-pleated sheet-like structure, while proline is present in a very small amount. So now we have discussed what we are doing nowadays. Now the primary and secondary structure of protein. In the primary structure, proteins are divided into the four categorized into four class: primary, secondary, tertiary, and quaternary, on the basis of the configuration and orientation in the space. In the primary structure, the structure is a linear structure. There is no any hydrogen bonds or covalent bond or van der Waals. Covalent bonds are present, but in between the amino acid. But hydrogen and ionic bonds are also, ionic interaction will be absent. Van der Waals forces will be absent. The example include insulin. Now in a secondary structure, the secondary structure, it is a second structure of the protein. It is a folding of protein in space with the help of a hydrogen bond, ionic bond, and van der Waals forces. The secondary structure will be divided into two classes: alpha helix and beta pleated sheet. Now beta pleated sheet is why because in there is the chain of chain of amino acids which is piling over each other and form this sheet-like structure in a space and form a beta pleated sheet-like structure. Now we are discussing only on this alpha helix and beta pleated sheet. Now alpha helix is it is the most common secondary structure. It is stable structure because it is formed by stability of alpha helix is formed by intra-chain hydrogen bonds, and it is always a right-handed structure. And in one turn it will contain 3.6 amino acids. The intra-chain hydrogen bond, because it is present in chain, so this interchain hydrogen bond, this here amino acid, this will pull each other. This will pull each other and form an intrachain hydrogen bond. It is present but in first amino acid and the fourth amino acid of the chain, and form this like structure. Now the amino acid which are present in alpha helix, this will include methionine, glutamate, alanine, and leucine. Methionine, glutamate, alanine, and leucine. There are the certain amino acids which do not favor alpha helical structure because these amino acids are a beta amino acid having a branch at beta carbon, like this amino acid having a branch at the beta carbon. This amino acid will include isoleucine, valine, and threonine. This isoleucine, valine, and threonine. Isoleucines, valine, and threonine, when they are present, they are always destabilize this alpha helical structure. Now the amino acid which are when present in this alpha helical structure, they will destabilize or disorient this structure. It includes serine, aspartate, and aspargine. When they are present, they will be always visible. Proline is also proline is normally present in alpha helix, but when there it will destabilize structure in very small amount or in small deviation or a small configurational change. Now the glycine is not able to form alpha helix. It may be asked in your examinations. Now beta pleated sheet structure. In beta pleated sheet structure include a piling of amino acid chain one over each other. It will be a two type, parallel and anti-parallel. In parallel, the chain are parallel to each other. In anti-parallel, they are opposite to each other. In parallel, it include flavodoxin, and in anti-parallel, it include silk fibrin. This beta pleated structure is stabilized by interchain hydrogen bond. and this interchain hydrogen form is between the chain. Valine and isoleucines are most common amino acid which are found in this beta predated cis structure. Protein is present, but it is in very small amount. Thank you so much, students.